Her expertise includes NMR and other biophysical and computational tools for the study of dynamic and disordered proteins and their interactions, including characterizing dynamic complexes that are mediated by multivalent interactions. She has, over the years, since I knew her when she was an undergraduate at MIT, gone from strength to strength, initially PhD at Yale, and now playing an important leadership role in Canadian science at SickKids and the U of T. And it's a pleasure to be able to host Julie. My only regret, of course, is that we're not doing this in person. The title of her talk today will be Intrinsically Disordered Protein Phase Separation in Translation Regulation and Bioinformatic Developments. Thank you, Julie. You're good to go. Well, you were.
Thanks so much for the warm introduction and remembering our meeting many, many years ago. So it's with real pleasure that I accepted your invitation, and I'm looking forward to later today to engaging with people in your program. So I just want to start by acknowledging the people in my group whose work I'm going to be talking about. Graduate student Brian Tsang, joint postdoc with Louis K. Tae-Hoon Kim, a previous postdoc Alaji Ba, who's now a professor at Upstate in Syracuse, Robert Vernon, joint postdoc with Alan Moses, Eva Pritanashak, Andrew Chong, and Sai Hao, as well as some collaborations, particularly with Lu Yang Wang at SickKids, Nachum Sonnenberg at McGill, Louis K. at U of T, and Alan Moses at U of T. So let me start just by acknowledging that proteins exist in a continuum of states, and they can be divided into monomeric states, specific oligomeric states, and higher order assemblies, as well as by their dynamic properties, stable low energy ground states, more dynamic states, and highly dynamic or disordered states. And we can think about this whole matrix and interconversions actually between them, those transitions during function and potentially pathology. So the lower right half of this matrix involves intrinsically disordered proteins, IDPs, or intrinsically disordered protein regions, IDRs, that make up about a third of the human proteome. And these disordered regions are involved in more complex regulatory pathways and are involved in complex proteins that are associated with complex diseases. These regions have a unique sequence features that lead to different ways that physical forces act on them, obviously very different structural properties, and that give rise to different functions, and also evolutionary implications. So in particular, I'm going to focus on the fact that IDRs contain sites for post-translational modifications. So these transitions during biology can be thought of in terms of, in many ways, and some of them are structural transitions. So if we think about the proteome, it's organized with some fully folded proteins, some almost 100% disordered proteins, but mostly with proteins that are mixtures of folded and disordered regions. And transitions during biology could be disorder to order transitions, partial disorder to order transitions, or even order to disorder transitions. But today I want to focus on transitions that are phase transitions to and from higher order associated states and to states with different material properties. So we can start with a well-mixed solution of proteins that demixes or phase separates to yield solute enriched phases and solute deplete phases, in these cases liquid phases. And these liquid phases can mature to become more gel-like or even solid and fibrous. So phase separation is an important biophysical organizing principle and regulating principle in all of biology, also involved in biomaterial formation. And we're all very aware that lipid phase separation from water makes membranes around cells, nuclei, or many organelles. And then there's even lipid phase separation within membranes that make lipid rafts. But more recently, we've become aware that proteins, nucleic acids, and even complex sugars can be involved in phase separation from water in both inside the cell and outside the cell to form what are called biomolecular condensates that give rise to membraneless organelles, also cytoskeleton and other intracellular structures, and extracellular matrix and biomaterials like elastin and biofilms. I'm particularly interested in the role of phase separation in the formation of these intracellular membraneless organelles, and many of them are RNA processing bodies, such as the nucleolus, in the nucleus, and P bodies and stress granules in the cytoplasm. These bodies are composed of hundreds of different kinds of molecules, proteins, and RNA, often with different multiple phases with distinct compositions and material properties. So what are the interactions that give rise to these condensates? They're dynamic, multivalent protein and nucleic acid interactions, with multivalency really being key. And this can involve folded protein interaction domains that interact with disordered regions that contain linear sequence motifs, such as SH2 domains for phosphotyrosine containing motifs, or SUMO for SUMO interaction motifs, folded binding domains for RNA like RRN or KH domains, or DNA binding domains for DNA, or even folded domains for other folded domains and oligomerization domains. But they can also involve groups within IDRs that I'm more interested in, to other chemical groups in IDRs or transient structural elements, such as kink beta structure, or for RNA or DNA. And RNA and DNA can also interact and phase separate without protein. But what are the IDR, and IDR nucleic acid interactions that are important? And these usually involve what are called low complexity regions of proteins. These are regions that don't use the full amino acid alphabet, and they may contain repeated sequence motifs, such as RGG, RG repeat motifs. And the interactions, we can think about them as weak physical chemical interactions, hydrophobic interactions, hydrogen bonds, charge-charge, cation-pi, dipole-dipole. I'm particularly interested in pi-pi. And when you say pi interactions, most people immediately think about aromatics. And I'm smiling because I'm thinking about when I met Steve. I was involved as an undergraduate in a project thinking about pi interactions of aromatics. So I'm still working on it, Steve. And but in fact, pi interactions can involve any groups that have sp2 hybridized atoms, and the peptide backbone amide is the most common pi group in a protein. And they've also got numerous side chain groups that are involved, including the arginine guanidinium group. And the reason I'm interested in this is because if you consider pi interactions in this way, and you look at the sequences of low complexity IDRs that are implicated in phase separation, they have a very large percentage of these residues that have available pi groups, or small residues or proline that have exposed backbone pi groups, up to almost 100% of residues in some regions, like the C-terminal domain of RNA polymerase II. With these pi containing groups. So in order to learn more about pi groups, a number of years ago, we did a data mining exercise on the PDB. And the PDB has been a real source of information. As a group that studies IDRs, I did not expect it to be so valuable, but in fact, it's been incredibly valuable for us. And we have mined the PDB and looked and found that the most prevalent residues that have planar pi contacts, of course, two aromatics, phenylalanine and tyrosine, but then after that, the arginine guanidinium group and the glycine backbone. And we also found that planar pi contact frequency increases with distance from regular secondary structure and also with solvent contacts. And these are properties that are very similar to those of disordered regions, suggesting that pi interactions may play a larger role for IDRs than for folded proteins. And so using this, we took the sequence statistics for planar pi interactions and came up with a predictive algorithm called p-score for IDR-driven phase separation via planar pi contacts. And we trained it on a small set at that time of known phase separating proteins. And we found that within the human proteome, about 5% of proteins had a high p-score that would mean that they are predicted to phase separate. And the gene ontology, localization, function, and process terms that were associated with them were showed RNA granules and processing transcription factor complexes. So a major role of RNA processing. There was also biomaterials and the nuclear pore. But this major role of RNA processing is perhaps not surprising, as RNA is a polypi system. Using this bioinformatics tool, we could look at some interesting things related to post-translational modifications. And we found that the phase separation propensity score correlated with a number of post-translational modifications in an IDR for methylation, often on arginine, and for phosphorylation. Since that time, there have been a number of phase separation prediction methods, and we published a review in 2019 of what we call first generation predictors. And there have been some new predictive algorithms since that time. And with these different predictive algorithms, some proteins known to phase separate are predicted by some of the algorithms and some by none. The union of them is valuable because they use different interactions, but all of those first generation predictors, including p-score, underpredict. A recent predictor, fuzz drop, coming from Michela Vendriscolo and Monica Fluchstrider's group, estimates that 40% of the human proteome is involved in driving phase separation, with up to 80% involved in condensates at some point. So a large percentage of the human proteome is involved in phase separation. But we wanted to develop an algorithm that was more comprehensive in terms of the physical interactions. And again, we turn back to the PDB for some data mining and looking beyond non-local pi-pi interactions. We looked at hydrogen bonding, charge repulsion, beta strands or kinked beta strands like LARCs, which are these low complexity aromatic rich kinked beta segments that can form amyloid-like stacked beta sheet structures and can contribute inter and intra-chain pi-pi and H bonds. And also we looked at solvation. And so when we pulled all of these out and compared for our set of phase separating proteins versus the PDB or the human proteome, you could see that these were distinguishable. So non-local interaction sequences that are unlikely to self-satisfy are important, as well as disorder, high solvation, extended structure, and this kinked beta. And we took 16 different interactions, and some of them correlated positively with our ability to train a phase separation predictor, and some negatively. And then when we flip the signs and then competitively trained with all of them, taking the top 8, we see long-range hydrogen bonds, long-range pi-pi, disorder, kinked beta, and electrostatics as being important. And so this 8-component predictor that we trained, we use actually 3 negative sets. The PDB is a common negative set, but it turns out the PDB has phase separating sequences in it. So it's not a true negative. The human proteome is clearly not a true negative, but we trained against both of those and a combination of those. So we actually have 3 different predictors. And if we compare to other predictors, we can see this does pretty well, even against this machine learning predictor, PS predictor. But our goal is not a black box machine learning algorithm, but really to understand the physical interpretability. What are the physical interactions that drive phase separation? And so we see this importance of solvation, disorder, pi, and kinked beta. And, thinking about moving towards improving predictions, there are single site mutants or alternative splice isoforms that are associated with disease, but the difference in the scores of many of these predictors to wild type are not really significant. And the other thing is most of these predictors, all of these predictors are focused on a single protein, but inside the cell, you've got complex coacervation with multiple proteins and nucleic acids. And also these predictors don't take into account post-translational modifications. So there's a lot more to do. So stay tuned for that. So I want to turn now from talking about the biophysics that underlies phase separation to talking about how the cell takes advantage of that to regulate biochemistry. And there's an increasing evidence that points to the functional roles of phase separation in the cell, obviously in organization, compartmentalization, in concentrating components. There's been interesting data related to its role in noise buffering of protein concentrations. Also the creation of unique solvent environments that can control binding, catalysis, and the energetics of folded protein conformational equilibria. But in terms of the processes that could be regulated, translational regulation is one significant function that has been described, and in particular, local translation regulation that's important for all cells, but in particular for cells that have unusual morphologies like neurons with long projections, also sperm. So neurological disorders may reflect a hypersensitivity to local translational regulation due to phase separation changes. And so we wanted to study this, and we generated what I like to think of as a model mRNA transport granule or neuronal granule. And we included in this 2 RNA binding proteins that are have disordered low complexity C-terminal tails that are predicted to phase separate, FMRP and caprin-1. These tails have RGG-RG motifs that can be involved in dynamic protein and RNA interactions. And both of these regions in isolation in our hands phase separate readily with RNA. And we can look in the NMR and see that these regions are disordered as we would expect in the mixed state, but they're also remain disordered in the condensed phase separated state with NMR amide proton chemical shifts showing narrow chemical shift dispersion that's consistent with the liquid nature of the condensed state and very rapidly exchanging weak multivalent interactions. And similarly, the caprin C-terminal low complexity region also remains disordered in the condensed phase. So going back to the PDB again, how can we understand what are the interactions in a very dynamic, disordered state? So we can take some snapshots from the PDB of arginine and glycine residues and think about what are the interactions that we might have dynamic exchange. And you can see pi-pi interactions and backbone hydrogen bonds, and even interactions with RNA from the PDB that you can see hydrogen bonds and electrostatic interactions. So dynamic interactions doesn't mean there's no structure. There's just dynamic structure, and it's useful to have these pictures to think about what are the interactions that are being sampled. Okay, so these two proteins are in related biochemical pathways affecting what's known as synaptic plasticity, really important for learning and memory. They're highly expressed in the brain. They're functionally involved in mRNA transport and stability and translation. And mutations in both of these proteins are linked to autism spectrum disorder and Fragile X syndrome, with knockout and loss of function leading to neurodevelopmental behaviors that recapitulate these disease in mouse models. These two proteins are co-localized in cells, and in particular in these membraneless RNA bodies, P-bodies, stress granules, but of more importance are, to me, are these neuronal mRNA transport granules that take RNA that's formed in the cell body of a neuron in a translationally repressed state out to the synapse. And when they arrive at the synapse, upon activation, receiving an activation potential stimulus across the synapse, they transiently disassemble. The RNA becomes available for translation. The proteins that are made are, in fact, can be considered learning and memory, because what they do, they can be cytoskeletal proteins that increase the size of the synapse contacts, or they can be for increasing the number of receptors at the synapse. And then immediately after making that protein, these granules reassemble. So we hypothesize that something about the synaptic potential controls the critical concentration for phase separation, and that enables the reversible assembly and disassembly of these neuronal granules to regulate activity-dependent translation. And we wanted to test this hypothesis by studying this neuronal granule model. So the C-terminal low complexity regions of FMRP and Caprin-1 are sufficient and necessary for phase separation of the full-length protein. And you can see that with these droplets that are formed for the full length and for the C-terminal, but not for deleting the low complexity C-terminal region. Both of those regions also mediate RNA binding at the level of hundreds of nanomolar. And we often have been using this small synthetic SC1 RNA, which is a G quadruplex forming RNA. But in particular, here's some data from the FMRP LCR that it binds non-specifically to any RNA. So what about synaptic stimulation could change the critical concentration for phase separation? And it's known in the literature that multi-site serine threonine phosphorylation of FMRP by casein kinase 2 represses translation. And then after stimulation of the neuronal receptor mGlu-R, there's the protein phosphatase 2A activity that dephosphorylates FMRP is induced, and that initiates translation. So in order to recapitulate that in vitro, we phosphorylated the FMRP low complexity region with casein kinase 2, and you can see in this mass spec that we have 8 to 10 phosphate groups. And then we can look at a phase diagram as a function of RNA and the FMRP LCR, and we can see that in the non-phosphorylated case, we need more protein and RNA to get phase separation, these droplets, than when we have highly phosphorylated. And that is that's consistent with theory, which says that electrostatics is really important for phase separation, with charge blockiness being important, so having positive and negative charge blocks. And here you can see that phosphorylation increases negative charge blocks. This is a sliding 10 residue window of net charge, and that is consistent then with the phosphorylation of FMRP enhancing its phase separation. Another post-translational modification that's seen on RGGRG motifs is arginine methylation. In this case, asymmetric dimethyl arginine modification is very common with protein arginine methyltransferase 1, PRMT1. And we can also generate arginine methylated FMRP LCR, and you can see with this mass spec adding 12 to 16 methyls on 6 to 8 arginines. And we see that the RNA binding activity affinity is not affected by arginine methylation, which was somewhat surprising, but you can see that phase separation is dramatically reduced. Similarly, moving from arginine to lysine dramatically reduces phase separation, but doesn't affect RNA binding. And this points to not just the positive charge being important, but the pi interaction of the arginine guanidinium being important in phase separation. So here's the test of the hypothesis of the role of phase separation in translational regulation. So we used a rapid reticulocyte lysate translation assay and luciferase mRNA, so we could use luminescence as a readout of translational activity. And here I'm plotting the inhibition of translational activity by either adding a standard translation inhibitor, puromycin, or the FMRP low complexity region. And what you see on this log scale is that the puromycin is sort of more linear, but with FMRP, you basically have no effect until boom, all of a sudden it's almost all inhibited. And with phosphorylated FMRP LCR, we need less protein, and with arginine methylated, we need more protein to inhibit translation. And if you take some of the assay out and you look in the microscope, where you have no inhibition, with the exception of some crud from the lysate, the well is clear, but where you have inhibition, you see droplets. So there's a strong correlation between phase separation and translational inhibition. And so this is our model then, with a temporal match of FMRP phosphorylation states with disassembly and reassembly of neuronal mRNA granules, and correlation between translational inhibition and FMRP phase separation, as well as the known phase separation of other proteins that are linked to synaptic plasticity, really supporting this idea that phase separation of RNA binding proteins underlies the assembly and disassembly of neuronal RNA granules and activity-dependent translational regulation. So we don't know as much about the role of arginine methylation in terms of regulation. Perhaps it sets a set point, but certainly with regulation by phosphorylation, we can understand that. And we're also considering the role of calcium influx through the receptors as an ionic strength effects on this disassembly and assembly. But the regulation of translation is not just about the translation enzyme mechanism itself. It's also about local mRNA stability. And caprin-1 has been associated with deadenylation, which is linked to degradation of mRNA. So we want to add caprin-1 in, and we see that FMRP and caprin-1 LCRs phase separate in a manner that's dependent on the phosphorylation of the FMRP LCR. And we can ask, what are the interactions that govern this co-phase separation? And turning to NMR, this is a C13N15 CON experiment. Because the NH is got a very reduced dispersion, we're using the increased dispersion of C13 to get more resolution. And what you can see is in green, the condensed FMRP phase that we've added caprin-1 into. And here we've got a condensed phospho-FMRP phase that we've added caprin-1 into. And interestingly, even though caprin-1 doesn't bind to non-phospho-FMRP in a typical bimolecular manner, while it does bind to phospho-FMRP, caprin can partition into either of the non-phospho or phospho-FMRP phases. So partitioning and enriching in condensates is different from bimolecular interactions. And that's something really important to consider. But what's also important here now is to look at the fact that there was broadening of many of the resonances in the condensed phases, and there are differential broadening in the two phases, the phospho and the non-phospho FMRP, and these include a number of resonances, but in particular, arginines and tyrosines and phenylalanines really broaden out. These pi-containing groups. And we can look at broadening with this intensity ratio, looking at the intensities in the condensed phases divided by the intensities of caprin just in buffer. And we can see that there's this significant dips to very low intensities across much of the protein sequence. But in particular, we have these arginine-rich regions where there are differences in the intensities between the non-phospho and phospho FMRP condensed environments. And this is likely due to differential charge interactions with the phosphate and the arginines. Whereas with the aromatic-rich regions, we don't have such differential interactions. It's very highly broadened in both cases. And this is just a difference plot. And so arginine, these aromatic regions, and tyrosine in those, tyrosine and phenylalanine in the aromatic regions have this pi character. So underscoring the importance of these pi interactions. So what about the tyrosines in those aromatic regions? They're known to be tyrosine phosphorylated, and we can phosphorylate again in vitro using EPH4A kinase. And we see that under certain lower concentration conditions, both being non-phospho and both being phosphorylated don't give rise to phase separation, but with non-phosphocaprin and phosphoserine threonine FMRP, we get droplets, and non-phospho FMRP, but phosphotyrosine caprin, we get droplets, and this is likely due to charge repulsion. We can do some bioinformatics with p-score, and we can see that as the number of tyrosine phosphorylation sites increases in the human proteome, we have a higher propensity of those proteins to phase separate, and also a higher association of those proteins with mRNA stability and translational regulation. So when we add SC1 RNA to our serine threonine phosphorylated FMRP and non-phosphorylated caprin droplets, we see the formation of multi-phase droplets. We don't see that in this other swapped phosphorylation system. And we can look at this as some kind of a logic gate. So it suggests how integrating different signaling pathways could control translation and RNA processing within cytoplasmic bodies. So these multi-phase droplets are liquid-like our single phase droplets, and both the phospho FMRP phase, which is this purple outer phase, and the caprin-1 inner phase, you can see these droplets fusing over time. And these dynamic liquid phases are also very similar morphologically to what's observed in high-resolution imaging of cellular condensates like stress granules or nucleoli. We don't have as high resolution images of neuronal transport granules, but if you squint, you can see that FMRP and caprin-1 don't actually directly colocalize. And maybe it looks like caprin is on top of FMRP. Okay, so what about deadenylation? Now that we've got these caprin condensates, we can look at the effect of condensates on deadenylation, and we use a 38A RNA substrate and the C0.7 catalytic unit of the CCR4 NOT deadenylase complex, and a gel assay to see the degradation over time. And what we can see as a function with buffer as a function of time, we have significant deadenylation, but inside our multi-phase condensate, the phospho-FMRP caprin-1 condensate with CNOT7, we see much more rapid enzymatic activity. That is not the case in this other condensate. And we can watch over time labeling the RNA and watch that the inner droplet actually disappears as the RNA is degraded in that subcompartment. We can also look at deadenylation within just a caprin condensate, because that's the inner condensate is caprin. So just looking at caprin, by ITC, initially we see that CNOT7 binds to caprin, and then we have a second phase of a thermal phase here of phase separation. And at these molar ratios, we see the rates of deadenylation buffer are the same as with binding, and we don't see significant phase separation, but here, where we have phase separation, that's where the rate is enhanced. So deadenylation rates are accelerated not by binding to caprin, but by being within a phase separated caprin environment. And we can turn back to the PDB to get some insights as to why this is. We look at the CNOT7 crystal structure, and we see that there's a C-terminal regulatory tail where there are two glutamic acids that coordinate magnesium ions that block the active site that would require this tail to be removed for substrate RNA to bind and be catalyzed. So I'm just showing some preliminary data here where we generated a deletion of this tail, where we call CNOT7X, and we looked at the catalytic parameters of CNOT7 in buffer, CNOT7X in buffer, and CNOT7 in the caprin condensate. And what we see is that CNOT7X in buffer looks like the catalytic rates of CNOT7 within the caprin condensed phase. So this suggests a mechanism for enzymatic regulation where you shift the conformational equilibrium between buffer or just a simple bimolecular binding to something that's different within the caprin condensate, where we open up this tail. So we're doing some more studies and NMR studies to further pursue this. But what about the mechanism of translational regulation by phase separation? And this is more simple to understand. It's just partitioning. If you have all of the reaction machinery in a single pot, and then you divide the reaction machinery into different pots, then you're going to be inhibiting it. And so what we have is differential partitioning of the translational machinery, including differential partitioning of the substrate RNA. So here we've labeled the RNA, which is in this inner phase, and here we've labeled eukaryotic initiation factor 4E, or cap binding protein, and we can see that that is primarily in the outer phospho-FMRP phase of these multi-phase condensates. So that's one example of pulling the translation machinery away from the substrate, but we're now doing a more comprehensive screening and trying to figure out where all the translational machinery is. So we can do our deadenylation and translation assays as a function of composition, FMRP, caprin, as well as post-translational modification of FMRP and caprin. And what we see is that we have for deadenylation about an order of magnitude regulation from buffer to these multi-phase condensates. With translation, we see on this log scale, we have 10 to the 4-fold inhibition in these multi-phase condensates relative to buffer. And so we can see then that phosphorylation acts as a switch that controls the phase behavior. It controls the multi-phase behavior, subcompartmentalization, and it controls enzymatic activity. So what in general can we infer about the impact of IDR phase separation on biology and disease? So I want to go back now. Excuse me. I want to go back to some bioinformatics and thinking about the way the proteome is organized. Again, about 60% of humans in the proteome have significant IDRs, and 20% have IDRs that make up half or more of the protein. And if we think about the energy landscape of IDRs, they can be involved in discrete complexes and liquid condensates and gel condensates, amyloid or glassy states. And then thinking about how mutations in IDRs could change this. So there are a lot of mutations in IDRs in disease-associated proteins, but these mutations are frequently neglected, and they're often annotated as variants of unknown significance because there's a lack of understanding of the pathological mechanisms. And many of the tools rely on positional sequence alignments, which work very well for folded proteins that have ordered tertiary structure. But these don't work very well for IDRs. But so we can think then about the variety of IDR mutations having biophysical impacts in changing phase separation thresholds, critical concentrations, or changing physical properties from liquid to gel to fiber, or changes in protein partitioning, exclusion, or enrichment inside condensates. So using p-score, our long-range planar pi contact driven predictor of phase separation that's not comprehensive, we see that proteins that are associated with autism and cancer are much, much more likely to be predicted to phase separate than the average human protein. And this isn't surprising because phase separation increasingly has been associated with a variety of biochemical processes that are critical and where perturbations can lead to disease and have been associated with perturbations that are linked to autism and cancer. And these start from chromatin condensation, as well as transcriptional activation, elongation, splicing condensates, then packaging into neuronal transport condensates, as well as the transport, and then once getting there, activity-dependent translation that we've talked about, synapse organization with a postsynaptic density for cancer, signaling receptor clusters, and activation of receptors have been associated with phase separation, and then actually signaling puncta that take information from various parts of the cell back to the nucleus are also understood now to be condensates. So there's a lot of condensate biology that's really critical in much of what we think about as core processes in biology. So what I was saying before is that a lot of the bioinformatics tools for understanding mutations in disease rely on positional alignment. So what can we do in order to push forward an understanding of mutations in IDRs or IDRs in general from a bioinformatics perspective? So it's really there really has to be a change in understanding what it means to be conserved, because positional alignability is often used as the definition of conservation of protein sequences in various organisms. And this only works positionally if you're talking about folded regions of proteins. So you could have IDRs that fold upon binding to something, or have folding upon post-translational modifications. And those IDRs can be positionally alignable because there's some part of biology which requires a folded state. But for IDRs that function in particular in phase separation, these are not necessarily going to be positionally alignable. So what we need then is, instead of thinking about positional alignment, we need to think about features. And so in collaboration with Alan Moses, we've been developing a bioinformatics of IDPs that relies on features. And here is a figure from a 2019 paper where we looked at the conservation of IDR features across the gazillions of years of yeast evolution in yeast IDRs. And this study used an approach of a null hypothesis for evolution of IDRs that was biased to maintain short linear interaction motifs, which we have since changed. But in this biased way, what we with this biased tool, we found that yeast IDRs were conserved in terms of their presence of their features at quite significant levels. So this is a percentage of IDRs that are significant in their conservation. And if you look at these features, we have amino acid content, we have charge properties, we have a number of biophysical, physical chemical properties, like for instance, including phase behavior. And then here we have repeats, elements, and complexities. Here we have the short linear interaction motifs. But most of these highly conserved properties reflect what I like to think about as polymer properties of IDRs, as well as properties that are known to be associated with phase separation, including aromatic and charge patterning, including some of the presence of these repeated low complexity elements. So using these features, one can cluster all of the yeast IDRs, and these clusters, many of them are related to biological condensates. So we see those that are that have Go annotations connected to ribosome biogenesis, which is related to the nucleolus. And then we have, here's another one, and DNA repair, which is related to DNA repair foci, and the nuclear pore, which is a condensate. And we have cytoplasmic stress granules, transcription. So what we see is many of these are related to phase separation. And we also can use the features to link to other functions. And it particularly, we tested the ability to use these features to localize proteins to mitochondria in the absence of a recognizable targeting sequence. And we've now extended this methodology to human proteins for IDR features. And as an example, we've got insights into nuclear and versus cytoplasmic localization in the absence of recognizable NLS or nuclear localization or nuclear export sequences. So these features can be related to localization, and more in general, we can think about them leading to enrichment in various biological condensates. And now we're moving to try and use changes in these features by disease-associated mutations to help us to understand how these disease mutations lead to pathology. So at this point, I'm going to stop and re-acknowledge all the people in my group who did all of this work, and my collaborators, including Alan Moses and Steve Scher for some of the bioinformatics work that I talked about at the end, and funding sources. And I'm very happy to answer questions. So I'm just going to leave this slide back up.
Thank you, Julie. Jean, are you going to jump on?
Can I ask a question here?
Yeah, please, Stephen. I thought, yes, please go ahead.
Thank you, Julie, for the wonderful talk. So I have a question regarding the process of phase separation. So is that a random collision effect or actively facilitated process, for example, mediated by molecular chaperones?
So phase separation is not mediated by chaperones, but when you say active, there are potential active events. So phase separation is a physics-based phenomenon, and is just due to multivalent dynamic interactions, and it can occur in vitro, in very simple systems, and organic polymer chemists use phase separation to make materials that do all kinds of useful things for us in our society. But in the cell, there are definitely active enzymatic processes that can be involved in regulating phase separation in both directions, in terms of facilitating it, or in terms of disassembling these biomolecular condensates. And chaperones do play a role in that, and they can play a role, but it's not required in general. But for stress granules in particular, there have been a number of suggestions for the role of chaperones in that particular biomolecular condensate. But it's not general, I would say.
Thank you.
Thank you, Stephen. We have a couple of questions on the chat from Chenghua Xiao. So he asks what, oh, Jean, good to see you.
Yes, so sorry, Julie, that was a great talk. I'm so glad to see you. I have a 1 p.m. that I have to hop on to, Stephen. So
okay.
I'm going to let you continue. And Julie,
Great.
maybe we can catch up later.
Okay, look forward.
Thank you.
Bye-bye.
Excellent. Thank you, Jean.
Bye-bye.
Chenghua Xiao asks, what phase would a protein crystal considered to be?
What phase? Okay.
Yeah.
It's a solid, right?
Mm-hmm.
A crystal is a solid, I would say. I mean, it's got hydration, but it's a solid.
But it's ordered.
Yeah.
That's the key thing.
Yeah.
How do you have a sense of how much water is in a phase separated condensate?
Yes, we do. We've actually measured using NMR amount of water in various condensates, and other people have done this as well. So it depends on the protein and on the conditions, the composition of the condensate, but anywhere from 60 to 70% of the water is there, so it's depleted.
So 60 to 70% of
what
the volume is water.
No. It's so we're So if you think about a dilute phase of as being roughly 100%, it could be 60 to 70%.
Well, a protein crystal on average is 50% water, 50% protein. Do you have a?
So it's more. So there's much more water. So for
okay, all right.
50, the condensate is closer to 70, and so it's intermediate between a dilute solution and a crystal.
Okay, and then so at the extreme, there is a small number of crystals that have only that have 70% water, but those are pretty rare, I think. And then the Cheng also asked, what is the biological interpretation for the excuse me, for pi contacts with high level association in phase separation?
What is the biological?
What is the biological interpretation for the role of the pi contacts? I think.
I mean,
there may not be one.
I think biology exploits physics. So, you know, whatever is around to be used. But actually, you know, in terms of, I think there maybe is something that I can say here. The pi interactions, you know, our predictor looks at planar pi contacts, but pi interactions have this fluidity of the energetic landscape, and this is getting back to some things that you and I thought about many years
ago. Exactly.
That you know, you can have stacked, or you can have these
the T-shape, yeah,
interactions, and there's a fluidity, there's a pathway, right? And so if you think about a lot of interactions in biology, like hydrogen bonds, if you move to a certain angle too sharp, they will break. Whereas with pi interactions, there's this very robust landscape over a lot of different twisting and angular orientations, where you have still quite significant energetics to play with. So I think that biology is exploiting pi interactions because of its geometric robustness in liquid dynamic phases.
Fascinating. Aishwarya Ranganathan asks if you think there would be a way of predicting the strengths of the interaction in addition to the propensity for disorder.
So I would say that quantification of phase separation propensity via these tools is a long way off. One of the reasons is that the databases are basically yes or no. Do you see a condensate under certain conditions or not? What is needed is like a high throughput experiment, modulating all kinds of conditions and modulating the sequence so that you can actually get more quantification in these measures, rather than it being binary prediction.
Sounds like a good case for a structural genomics program for IDPs, doesn't it? I'm afraid we're going to have to draw this to a close so we could give Julie a break before she starts with the graduate students at 1:15. I want to thank you again for visiting the Institute for Quantitative Biomedicine here at Rutgers virtually. And again, I regret that we weren't able to entertain you in person. Well, congratulations. That was a tour de force of a presentation.
Thanks, Steve.